Bismillahirrahmanirrahim. Another video on the November 22 paper 4.1, and in this we're going to be discussing question 4, question 5 and question 6. Now we start with question number 4. Adenosine deaminase ADA deficiency is an immune system disorder caused by a recessive autosomal mutation. You know, autosome means the body chromosomes, not the sex chromosome, which is the 23rd pair. So they are on one of the 22 pairs which control the body characters. So those are called autosomes. Severe combined immunodeficiency caused by a lack of AD is called AD-SCID, SCID. Genetic engineering is used to make a recombinant human protein to treat people with ADA, SCID, AD-SCID. Outline the principles of genetic engineering. Now that's a very direct question. It doesn't require any ambiguity, and these are the direct questions in which you should be able to score marks. Because if you've done 3 years past paper, all the variants, well, you would have found this in some other past paper as well. So you need to memorize that, you need to ratify that. So add DNA to give a new protein, restriction enzymes to obtain the gene, or we can say restriction enzyme to cut the plasmid, then combine the gene into the vector plasmid, then introduce this recombinant vector or plasmid to the bacteria, and then clone these and recombinant organisms, and they will produce that protein for us. So outline the principles of genetic engineering. So outline means you're just going to give me a brief outline of it, not the detailed of it. A very simplified version which you can now memorize. Add the DNA to give a new protein. You add a new DNA, the gene or the DNA, to make that protein into some other organism. So you use restriction enzymes to cut the DNA or the gene from the organism from which you are for which you are producing the, like for human insulin, you'd cut the DNA from a human chromosome where that insulin gene is present. Or you use a restriction enzyme to cut the plasmid. Then you combine the gene with the plasmid, and then you introduce the recombinant plasmid into bacteria, and then you clone these bacteria because they will produce insulin for you or the desired protein for you, whichever you need, where the deficiency, where the person is suffering from a deficiency of that protein. Now coming to the B part of the question. In 2016, gene therapy to cure ADS-SCID was approved in Europe. The gene therapy involves 3 main steps. Blood hematopoietic stem cells are taken from the bone marrow of the person with ADS-SCID. The functional gene and its promoter are inserted into the blood stem cells. A single fusion injection of the gene corrected cells is given to the patient. Explain why a single infusion of gene corrected stem cells is enough to cure the disease. Very easy. Stem cells will divide or multiply, will make new cells, and because the correct gene is inherited, will passed on to be present in the new cells. So the problem will be solved. The stem cells will divide, multiply, and the correct gene will be passed on to the new cells. So the person will not suffer from the disease because now that protein will be made. Some cells divide, make new cells, gene is passed on to the new cells. So there were only 2 marks and 2 points which we could have written for this. To the part 3 of the question, explain why a promoter has to be transferred as well as the desired gene. You see, the promoters are where they're recognized by RNA polymerase to switch the gene on, so that then RNA polymerase can allow transcription to take place at the right time, and this should be done at the right time in sufficient quantities so that to allow the binding of RNA polymerase. So promoter is the region which is just an area before the gene. Just like you have in your books, before the book starts, you have an introduction and then a preface and all that, and the contents, and then the book starts after a few pages. Because first is some sort of an introduction, and then there are the contents, which may be on 2 pages or 3 pages, and then the actual book starts. So the promoter is the area before the gene. It is also made up of nucleotides. It's a portion of the DNA, which is called the promoter, which is recognized by RNA polymerase. So explain why a promoter has to be transferred as well as the desired gene. So the promoter is there needed to switch the gene on, and it should be turned on at the right time so that it should be produced in sufficient quantities and then to allow the binding of the RNA polymerase. Now coming to part 3 of the question. A modified retrovirus is used to insert the new gene into the DNA of the blood stem cells. State two ethical considerations of using a retrovirus for gene therapy. So a modified retroviruses are those which have the RNA. So a modified retrovirus is used to insert. So we use that as a vector to carry the gene into the cell. So now retroviruses, we change its RNA. So a modified retrovirus is used to insert the new gene into the DNA of the blood stem cells. State two ethical considerations. Yeah, retroviruses must not insert in the wrong place. Should not cause cancer, should not cause or start an infectious disease or an infection, and it must not cause an immune or an allergic response because naturally we react to viruses. Look at us how we react to the coronavirus. So that is what we had to think. So the ethical considerations that this virus should not insert in the wrong place, should not cause cancer, should not cause an infection or some disease, and should not cause an allergic or an immune response. So the retrovirus must not insert in the wrong place, or we can have said this virus must not cause some sort of a cancer, or we could say must not cause some disease or an infection, or you could have said must not cause some sort of an allergic response. So ethical consideration is that the viruses, you see, what do we know about viruses? We just know it as an alarm bells ring when we talk of viruses, because we talk of viruses and we think of all the viral diseases which human beings suffer from. Polio, COVID, coronavirus, which was the most lethal in the last 3 years that we have been seeing it. And then the flu, the influenza that we have is also a viral disease. So we do have a lot of viral diseases and we hear a lot about it. And so we usually do not consider the virus as the good guy. It's considered as the bad guy, which is going to cause some sort of issues. Okay, now coming to the last part of this question. Part C, the gene therapy techniques used to cure ADA-SCID is not suitable for treating the genetic disease called Huntington's disease. A newer technique called gene editing could potentially be used instead to cure Huntington's disease. Explain why gene editing is more suitable as a potential cure for Huntington disease. Well, what is Huntington's disease? HD. We just sort of abbreviate it as HD. HD is caused by a dominant allele, and adding a normal recessive allele would not work. Gene editing will be need to delete or inactivate the HD or the mutant allele or the DNA. And because it has the surplus CAG repeats, so if we could somehow remove those, then those could be, because the repeats is called a stutter, and the more the stutter, the early the onset of the disease. So in gene editing, we would somehow remove that DNA. Gene editing means deleting portions of DNA or adding, deleting or adding, just like you do editing in an article. You remove something and you write something else. So this is what you had to write for these 3 marks. Difficult question for you to think of, but then if you knew what was HD, then you could have come up with it. So if you just wrote HD is caused by a dominant allele, so you could have got a mark for that. HD caused by a dominant allele. So if it is a dominant allele, it means is H is the disease or H small h is the disease. Now, adding a normal recessive allele would not work. If you added the normal recessive allele, it would not work. People who are normal are this. So, gene editing can delete the DNA, or we can remove the stutter. You see, in the end, what we have is we have a lot of CAGs, cytosine, adenine, and guanine repeats. It comes again and again. More the repeats, early the onset of the disease. So if we could edit this portion, I mean remove this portion, delete this part of the DNA, then we could maybe find a cure for it. Now, coming to question number 5, a large number of alpine plant species grow in the mountains of New Zealand, South Island. Alpine plants are defined as plants that live above the tree line. Like we see a lot of alpine plants in Murray when we go in Pakistan, which is the height above which trees cannot grow. The current tree line for most of the South Island is 1200 meters. The alpine areas nearest to South Island are 1500 to 2000 kilometer away across the sea. Many of the South Island's alpine species live nowhere else in the world. Explain how a large number of alpine plant species developed on South Island. Now, this is a very general question, and we've got to see and give an answer which is, of course, very befitting. And it's a 5 mark. So, how a large number of alpine plant species developed on South Island? Now, such a question has a very large marking scheme. And when you look at it, it says evolution, either speciation or natural selection. Then it is very random and spontaneous, and mutations occurred. And adapting some plants to alpine habitats or high altitude. Then these plants survived. Then selection pressures, they were able to withstand the cold and the strong winds and the thin soil. Then alleles for the alpine features pass to the offsprings. Then change in the allele frequency. Population becomes morphologically and physiologically different, and less herbivores, so less competition in this new niche. So there's not many animals which eat those, or there's not many weather adverse conditions which is going to result in the small trees really actually surviving. And so this is large number of alpine plant species. So the fact that they survived, that means that they're very well adapted to this new environment. Now you can see there's something like 10 points, and out of these 10 points, you could have written any 5 to get the 5 out of 5. So whenever there's a question on evolution or natural selection, some points you can write in nearly every question. Now, like natural selection, you can write it's a random event, then mutations, then adapting some plants to alpine habitats, these plants survived. Then selection pressure is cold and strong winds. Then alleles for alpine features passed to offspring. So the desirable alleles will be passed on to the offsprings. And this will result in a change in allele frequency. So these points you can write just without even thinking, and you can write in every question. Less competition in the new niche. That is why they're surviving, because there's less competition, and populations will result physiologically different. So this is for the 5 marks that you have to give me. So there are 10 points, but any 5 of these would have been fine. The B part of the question says figure 5.1 shows 2 aspects of the history of South Island over the last 3.9 million years ago. The dashed line shows how the mean height of mountains in the Clyde region of South Island increased over time. The mountains in this range have a mean height of 2,400 meters at the present time. The solid line models the height of the tree line over time based on geological climate data. The tree line was higher when the climate was warmer, and the tree line was lower when the climate was colder during ice ages. So present time and time before present in million years. With reference to figure 5.1, identify with reasons the time period when South Island's alpine plant species developed. This question was a little difficult for students to interpret. Why? Because figure 5.1 proved to be difficult for candidates. The question was based on the idea that alpine plant species, they're not alpine trees, they're alpine plant species, can only begin to develop when the mountains are higher than the tree line. So the correct interpretation of figure 5.1 showed that the mountains were consistently higher than the tree line from approximately 1 million years ago. This point. Approximately 1 million years ago. And many candidates focused on the information that the current tree line is 1200 meters and used this to give a wrong time figure from the graph. So you see, you had to understand, it was a little difficult. It was a very difficult question. It wasn't an easy question. So you had to really figure this out. So as you can see how I'm wording this now, Time period is from 0.95 million years. You see, this was in million years to the present. But what has happened? The mountains are high enough to exceed the tree line. So mountains were only then high enough to exceed the tree line. And then this provides alpine habitat or niche. And then we come to the last part of the question. Suggest how DNA sequence data could be used to confirm the time period you identified in part one. Now, how will you do that? You compare, find similarities and differences in the DNA. Yeah, but what do you compare it with? Of alpine species and the lowland species. And find the time when species evolved. And many DNA or nucleotide differences or mutations means that they separated a long time ago and have a distant common ancestor. That's a very typical sentence that we use. If the differences are large, then it means they separated a long time ago and have a distant common ancestor. So all you have to do is that you have to compare the DNA of the alpine species and the lowland species. So whose DNA are we going to compare? So you would have got the 2 marks for that. Because it says suggest how DNA sequence data could use to confirm the time period. So you compare the DNA of alpine and lowland species and find the time when the species evolved. And many DNA differences were then mean many differences if they are present. That means they separated a long time ago, or you could say they have a distant common ancestor. Now coming to question number 6, selective breeding has been used to improve the characteristics of crop plants, including maize and rice. Outline how vigorous uniform varieties of maize were developed by selective breeding. So selective breeding, you see, what do? You cross the plants with the desired characteristics. Then you take the best offsprings and repeat over many generations. And this is of course inbreeding, or you can do hybridization or outbreeding. And this will give us hybrid vigor. And so 4 marks for that. And F1 have the same genotypes or the uniform phenotypes. Now, all we had to do is you had to talk about artificial selection. But as I read the exam report, many candidates describe points relating to artificial selection in general without focusing on the specific example of creating vigorous uniform varieties of maize. Most candidates were able to describe the crossing of plants with desired characteristics. Many of these needed to go on to express the idea of repeatedly selecting only the best of the offsprings to breed from. Strong responses included the idea of initial breeding to achieve uniformity and subsequent outbreeding to produce hybrid vigor. So if you can look at the points that I've written is breed plants with desired characteristics, breed best offsprings and repeat over many generations. Inbreeding may have resulted in depression, or you can get more uniformity when you do inbreeding, and then this leads to hybridization or outbreeding and hybrid vigor in the offsprings. Now coming to the B part of the question, IR8 is a variety of rice plant that was developed in the 1960s. IR8 was developed by two PETA PETA, which produces a high yield, this one, and DGWG, which is a dwarf variety. So this is the one which has got very short stems, and the PETA is the one which is very long stems. But the fact that it produces a very high yield, high yield means more of the rice, because but this one is a dwarf one. That means its stem is very short. So figure 6.1 is a diagram showing the 3 varieties of rice, and what we did is we developed this IR8. Now suggest why IR8 is an improvement on the PETA variety of rice. Why? Yes, because you see more of the energy is going to spend on the seed, and less energy is going to be used for the growth of the stem. And that is what was supposed to be interpreted. So what we have to understand is very few candidates were able to interpret the diagram to explain why IR8 was an improvement on PETA. Many candidates described better yield or more edible grains without expressing the idea of the grains constituting a greater proportion of the plant in IR8, which relatively with relatively less waste in stem and leaf growth. You see, in a rice crop, we are only interested in the rice seed. We are not interested in the stem or the leaves. So if less energy is spent on the stem and the leaves and more energy is spent on developing the fruit, which is the seeds, then the farmer gets a better crop yield and of course makes more money. So this is what you were supposed to interpret, that because of the short stems, they would be less likely to fall over. You see, when there is a wind, yesterday there was rain and the wheat crop was badly affected in Pakistan because of the wind. You know, some of the wheat which was mature had sort of collapsed and fell over. So that is going to destroy all the seed, all the fruit that we were going to get, all the wheat seeds that we were going to get. And the fact that it was high yielding, so we would have a higher proportion of the grain. But you have to write only one of these. I'm not saying you have to write both of these. So having a short stem means they're less likely to fall over when there's strong winds or rain or hail or storm. So it will survive that because they're short stemmed, dwarf. And of course, the fact that it had more crop yield, so it will have more proportion of the grain. And coming to part 2 of the question, in 2009, scientists produced a new variety of rice, IR64 sub 1, by breeding together FR13A, a variety which has a low yield but has an allele for flood tolerance, and IR64, a variety which produces a high yield. So we've got a high yield and we've got an allele for flood tolerance. The scientists sequenced the DNA of these three rice varieties. Suggest the benefit of sequencing the DNA of IR64 sub 1. Now, why are we sequencing the DNA? You want to check the alleles or you want to check the gene or the DNA. Does it have the sequence which is going to result in high yield and flood tolerance or not? So, I mean, we have to check it before we start sort of planting that seed and saying, okay, well, this is now flood tolerant and it is also high yielding. We can't experiment. So why did we do the DNA sequencing? We did it to check whether it was the DNA contained those two alleles. Check for alleles for high yield and flood tolerance. You could have even said gene, you could have even said DNA. So all this was correct. Gene, allele, DNA was correct. Okay, coming to part 3 of the question now. After a few generations of breeding, the scientists crossed IR64 sub 1 with IR64. Explain why the scientists crossed IR64 sub 1 with IR64. Why did we? To maintain the high yield or to give a higher yield and to avoid inbreeding depression so as to maintain hybrid vigor. So why do we do that? So that this, of course, doesn't lead to inbreeding depression. So to get hybrid vigor, we did that. So to maintain hybrid vigor and to maintain the high yield so that in the future generations, this would still be present. High yield and hybrid vigor will result in high yield. Part of the question. Thalecress arabidopsis thaliana is another plant that has dwarf varieties. Scientists treated three varieties of A. thaliana with gibberellin. The wild type, normal, non-dwarf variety, and the dwarf variety A, and the dwarf variety B. So two dwarf varieties were chosen, and the scientists treated three. One was the normal one was the dwarf variety A, and dwarf B. Figure 6.2 shows the responses of the three varieties to treatment with gibberellin. Wild type, normal growth after gibberellin treatment. Variety A, normal growth after gibberellin treatment, and variety B, normal growth after gibberellin treatment. Oh my God. So the B did not respond. The A has responded, and the normal one has also responded. Now it says with reference to figure 6.2. So this is now, let's look at it again. This is A and this is B. So with reference to figure 6.2, so I've got to refer to this. Suggest explanations for the different responses to gibberellin shown by the dwarf A and dwarf B. So I've got to talk about this one and I've got to talk about this one. Now, what has happened? In variety A, gibberellin binds to the receptor, fine, and the DELA protein is broken down, and the transcription factor is released, and there is resultant in stem internode elongation, or you can say growth has occurred. Well, you can see that. You can see this, that the growth has occurred. You can see this growth has occurred. We are only going to talk about variety A. So growth has occurred, and A is LE has no big LE alleles, does not produce its own active gibberellin. It does not produce its own active gibberellin because what did we do? We treated it with gibberellin. But what has happened in B variety? We treated that with gibberellin also. Does not respond to gibberellin and has non-functional gibberellin receptors because nothing happened to it after the gibberellin treatment. So it maybe doesn't have the receptors or did not respond to the effect from gibberellin, has non-functional gibberellin receptors. So this is what we had to understand is that the dwarf varieties were supposed to remain dwarf. But then when we applied gibberellin to them in A when we applied gibberellin, the gibberellin will bind to the receptor, DELA protein broken down, the transcription factor or the PIF released, and this is going to result in stem elongation or stem growth. And so this is how what we had to really answer this question. And this is with reference explanation 4 marks. So you have to give me explanations to that. You see, you have to realize that the dwarf varieties have an issue. The gibberellin, you see, when we added the gibberellin to it artificially, it binds to the receptor, the DELA protein breaks down, the PIF binds to the DNA, stem growth occurs. And it has small LE. So it's homozygous recessive. It does not have the dominant one, big L, small e. And A does not produce an active gibberellin. While in variety B, it just does not respond to the gibberellic acid. GA is for gibberellic acid or gibberellin, you can use the word that. And it lacks the GA receptor. You see, for every hormone or chemical growth regulator, you have receptors on which it puts, just like the active site of an enzyme. So if you say this is the receptor, and on this receptor will the GA fit on it. So if there are no receptors, so where will the GA fit on it? There are no such receptors present. Part 2, BZR1 is a transcription factor that helps to regulate growth and development in A. thaliana. Outline the features of a transcription factor, 2 marks. Now, transcription factors are what? They bind to the promoter and they switch the gene on and allows the binding of RNA polymerase to the DNA promoter. So BZR1 binds to promoter to switch the gene on and allows the binding of RNA polymerase to the DNA promoter. So basically you have to only give me 2 points, and this completes this question and this completes this video, and the rest of the questions will continue in the next video.